So now, at this point, we want to launch into what we consider the grassroots of rare diseases, and that's the basic research. And one of the themes of this year is defects in the endoplasmic reticulum and Golgi. And the endoplasmic reticulum is where proteins, many proteins that are defective, start their life. And a lot of what they have to do is to get organized. They got to find the right way to move to the place they need to go to. But first, they got to get in shape. And our first scientific speaker, Dr. Bill Balch from Scripps Research Institute, can come on up, and he's going to tell us about some really interesting studies that he's done on proteostasis. And I think the title of your talk is The Role of Proteostasis in Rare Disease.
Get back to the beginning here. All right. The former speaker decided to give my talk himself. Okay, we're back to the beginning here. What I'd like to do is take you from really basic science to a therapeutic that we think is going to be sort of a general way to begin to think about many of these rare diseases. And so I'm going to have to introduce some terms to you. For the kids in the audience, I'll have some worms and mice so that you can see some of those in action and see what some of these therapeutics that we're trying to develop will do. But really, most of the talk is geared toward the scientist, of course, to give you what we think is a new platform. And it's a new platform because this system, which I'll define in a minute, proteostasis, really addresses a common principle that is driving many rare disease, and that's a mutation. It's a problem in the folding. It doesn't want to put the protein that's driving the disease doesn't want to put itself together properly. And what we're learning is at the moment the system. What we're learning about is the system that teaches a protein to put itself together. We're also learning that we can manipulate the system, and we can make a protein that doesn't actually like to fold actually fold. And so keep in mind your own favorite disease. I'm going to talk about cystic fibrosis because we have a lot of effort, but in reality there's many things that apply directly. Many things you're going to hear apply directly to your own condition. So the first thing I have to say, and actually it emphasizes the introduction, is that this is largely a collaborative effort. You're going to see my colleagues Andy Dillon at Salk, Evan Powers at TSRI, Jeff Kelly at TSRI, Gerald Manning at Salk, Joe Gottesfeld at TSRI, Rick Muramoto, who's in the frigid Midwest, Northwestern, and John Yates at TSRI. We all work together to provide bits and pieces of the puzzle, and just as advocacy groups and whatnot have to come together to work to move a problem forward, we have the same issue. No one of us is the answer to getting at these types of diseases. Okay, so here's what we need to think about. And really, because I get to give the first talk, I kind of get to give an overview. We're getting lots of activity here, even from the lights. I want to provide some questions, and sort of the questions we worry about. Rare disease affects health span. And it actually, this rare disease usually affects a protein. And a protein is part of a much larger proteome. That is, you're full of proteins. And you have one protein that happens to be in the middle of this quagmire of proteins that just fails to function. It doesn't interface properly. It's a reject in a party. And as a consequence, you have disease. So what we're trying to do is really ask the question, just what's the relationship between this protein that's out of step and this proteome that is really responsible for normal function. So we're going to focus on this question of protein folding because it's universal. So what are the biological systems that manage healthy protein unfolding? After all, we want to be healthy, so there must be systems that keep a protein healthy. And we're going to initially focus on this class of proteins in a very general way. How is this program challenged by a rare disease, i.e. it's out of step? So what does that do to the system? And of course, what does the system try to do to protect you and counter that insult? After all, we don't die immediately from a misfolding protein. There's a system in place that tries to buffer that, to put it back into place. And if we can get that system working a little better, then it'll buffer it more effectively. And of course, then finally, how do we therapeutically address the problem? Either to maintain sort of a steady state, to reset the switch, to make it behave itself, and or sort of improve generally the health span so that the patient isn't suffering as much as a consequence of the disease. So here's what I'm going to talk about, and it's really about managing the yin and yang of what we call proteostasis or protein folding balance. And really, you're all familiar with yin and yang, and really the cell has the same problem. There's a balance on one side that wants to promote folding and keep things healthy. On the other side, when a protein has a mutation, it's taken out by the system, it's degraded. And the consequence of that is you lose that function. And that leads to organ failure and all the types of symptoms you have in different diseases. So what we're trying to do is adjust this yin and yang so that in disease, which tends to land on this side, we push it back over to this side to generate a more healthy state. Okay, so part one, I'm going to briefly go through building an understanding of what we call the proteostasis system, tell you what it is, and why it affects and helps us maintain health span. This is the work of my colleagues, Morimoto, Dillon, Kili, Evan Powers, and of course myself. Part two, I will try to help you get some insight into the balances and imbalances that occur in these rare diseases through protein misfolding. And also emphasize the power of systems approaches, genomics and proteomics especially, the work of John Yates, to address these challenges. And then what I'm going to end up with is a few slides which just illustrate how we're trying to move in a direction to restore this balance, taking advantage of what we call the emergent properties of the protein folding or proteostasis system to reset this yin and yang, and that is protect and improve health span. So what I'm talking about here is a general platform to begin to address these issues. So if we discover a drug that affects one rare disease, it actually means we should immediately test that in other rare diseases because they may actually have this same common problem. They all look different at the end, but they have the same common problem that starts and sets the disease in motion. So let's start with building the platform. This is really for the basic science. Guys, here, what we're going to do is start with an unfolded protein. This is the rare disease protein that triggers your disease. And what it has to do when the cell makes it, it's all disordered and all kind of flying all over the place. And it has to move down a folding pathway to become energetically stable. And this is the guy that does the work. And if you never make this point, if you're trapped up here or trapped here, you fail to perform the normal function and you're in trouble. You can kind of blow that up, and if you look at it, you can see trying to get through sort of this folding pathway that gives rise to that native state is a labyrinth of mountains and valleys. And as a matter of fact, there's lots of ways that a protein that's just been made in a cell can try to proceed down that pathway. But it usually turns out there's one preferred pathway as shown here. And in a rare disease, oftentimes the mutation takes out that preferred pathway. So for the most part, the protein tries to go down these other routes, but it gets in trouble because the system has quality control checkpoints. If you try to go down this route, the system knows there's a problem. But it turns out that we can probably modulate this pathway to give the protein more options. So even though you inherited the mutation, and this is what evolution has sort of optimized itself on, it turns out there's many options that we can work with. And the idea is to home in and understand those options. So what are the biological system that manages what I call folding chemistry by this polypeptide chain? So it's this term proteostasis. So what is it? So we call it the adaptable chemistry of the protein fold that's generated and maintained by the biology, the system or the network, to promote normal physiology. And what this actually does on a daily basis is help you manage stress, manages disease, and of course, it plays a major role in health span. And we're learning that actually the types of activities we're finding that improve health on a daily basis can actually make you live longer. So it's a kind of a double whammy if we can really get it to work. So central dogma says we go from DNA to RNA to function. This is the functional protein at the bottom of that energy well. And it's not that simple. When you have this unfolded polypeptide here, lots of things can happen. That is, you don't always achieve function. If you misfold in the context of a stress environment, you have an aberrant structure and you get cleared by the system. If you never approach a folded state, you're immediately removed by the system. On the other hand, as you all know, you've heard of Alzheimer's disease. If you don't fold properly, you can get an aggregated state, and that leads to many neurodegenerative diseases. So there's lots of possibilities. It's not straightforward to go from here to here. And the real question is what manages that. And there's a whole system. There's 2,000 components. It's like having 2,000 house care people come to your house every day, and one takes care of the dishes and the other takes care of the laundry. You know, it's the system set up to protect that protein and make it really reach a functional state. So, and I'm going to tell you about that system in a minute. The other thing, though, that we're learning is importantly, there's feedback. That is, all these pathways that are at this endpoint here feedback to DNA. And ultimately, that means we might be able to train the system to behave itself. And there's a lot of activity in this area. I'll talk a little bit about it in terms of the type of effort that we're pursuing. So, throughout life, we're dealing with normal physiology, but in the case of rare disease, pathophysiology, which is basically single nucleotide polymorphisms and mutations, as well as defects in transcriptional, translational, and post-translational events. We call these errors. They're initiated at birth, particularly in the case of rare disease, but sometimes they have a late onset because the protecting system keeps you in check. And as you age, it turns out we're learning this proteostasis system fails. Of course, we all know we get old. And we also know that that young athlete always beats us in that 10K marathon compared to our own efforts, right? And that's because this system that protects your muscles and everything doesn't keep you active enough to be competitive. And so we're really talking about protein maintenance here. Lots of players. These are structures of proteins. These are just examples of the many types of structures that play a crucial role in maintaining the proteome. We have chambers that actually refold proteins. We have chambers that remove unwanted proteins. And then we have factors that specifically bind that protect an individual protein at a different stage of its life. So when you think about all these sort of proteostasis components that are shown here, and you look at this energy landscape that I talked about, it turns out these guys are all specialists. They prevent aggregation, they help in misfolding, they help to prevent misfolding, they help guide the protein down the right path. We have some specialists, and they're turning out to be very important, that sit out at the end here, that are really sort of the managers of the endpoint, and really help a lot of proteins maintain proper function. And then, of course, we have all these degradation pathways that take out anybody who's a problem. And as a consequence of that, they protect the cell. They keep the cell nice and clean and healthy. There's another way to look at it. If we take that energy diagram and make a slice through it, we can see each protein goes through all these different folding steps. So it doesn't just go from here to here. It has to bounce through all these intermediates. And sometimes it goes down the wrong side and gets aggregated. And each of these components seem to manage a particular step. And the important thing, though, is what we're learning is this is reversible. If we end up over here, we can run it back up to here and give it a second try in a new environment, and perhaps that new environment that will support the fold. The system we're talking about is ancient. That is, these are a list of some of the components that we know about, and if you look down here in yeast, every one of these is found in a yeast that is found in a mammalian cell. What we know now, though, is that what happens as you begin to go from a single cell to a complex organismal state, such as a human, what you do is you expand the number of components within that class because the complexity of the protein fold increases. So, for instance, yeast doesn't have the rare diseases that we encounter in humans because it doesn't have the organismal complexity it has to deal with. But there are specialists that have come up to deal with these problems, and we're going to try to take advantage of these as specialists to make them work a little better to help the rare disease state. The other surprise when we started looking across tissues is that each one of these is a different tissue in your body. And this is just representative of 400 to 500 of these proteostasis components. And if you look here, what we're looking at is the black means an average level, red means a very high level, and green means the protein's almost non-existent. And what you see is that the composition of the proteostasis network in the brain, which is indicated by the fluctuations of red to green to black to red, is completely different than that which drives the immune system. So when you have a rare disease, you have a protein that's targeted to a particular type of tissue, and the system that protects it is actually specialized and designed to protect it. And that means we get some specificity out of this so that we can go in and try to readjust, for instance, the immune response in a way that really won't affect the brain. Of course, you don't want to have something that comes in and affects the whole body and creates a problem. So we're going to be taking advantage of this to therapeutically address folding problems. So we know that this composition you see here also changes during development, changes during aging, and is highly responsive to stress and disease. So this system that I'm talking about is the very system that protects you from all the nasty things that happen to you during life. And it has to be there because it's really working for you, and the environment is a major force to deal with. And of course, in rare disease, it's more of a problem because you're out of step. This is a little bit of a three-dimensional Einstein view of what we're thinking about. And really, a protein that has to be made has three problems. First of all, it has a folding rate, it has a misfolding rate, and it has what we call a thermodynamic stability. And if we plot each of these relationships on a three-dimensional state here, what we do is we can define a position in a network as to its relative stability with respect to the environment of the cell. Importantly, we can also define just where the protective boundary is in this cell. It's just like for kids, mom protecting the kids up to a certain point, and then they go off to high school and college, and they're out of control. And what that means is that this is the folding environment, and it's the protective environment that gives you function. If you get on this side of this boundary, you're unfolded, you're in a degradative environment, and you're in trouble. And this gives you loss of function, gain of toxic function disease, so you're in trouble. And that's what that looks like over here. Here's a rare disease protein. It's sitting over here. It's just out of reach. And what we want to do is be able to grab that protein and either pull it back in, or we want to move this boundary a little bit so it actually grabs it and protects you. Okay, how do you move the boundary? Well, it turns out the cell is full of pathways that are illustrated here. This is our sort of icon for a proteostasis network. And there's pathways that are defined in biology that include various epigenetic pathways, that include heat shock response, a variety of different approaches, which can grab the proteins and move them around and adjust them to affect a different state of proteostasis protection. What I'm going to do is just illustrate for you the IGF-1R pathway and the dietary restriction pathways. And that's shown blown up here. And that is that we know through the work of Dillon and Kelly that you can manage the health span and longevity through manipulating this pathway. So for instance, this is an IGF-1R signaling pathway that works through specific transcription factors. This is a dietary restriction pathway, which if you eat too many McDonald's hamburgers, you're in trouble. This is a mitochondrial pathway. But the fact is, what we can do is we can test the utility of these pathways in various models. So here's a worm model. Well, the kids are still here. This is a worm that has Alzheimer's disease, but it has Alzheimer's disease of the muscle. This is generated by the Dillon and Kelly groups. And the fact is, it's frozen. It can't move around because its muscles are paralyzed. Here's what we did. Here's what the Dillon and Kelly group did, is they changed this pathway, and they restored the mobility of the worm. It actually lives longer, and it lacks the quote-unquote neurodegenerative disease. They moved on to show that this is an Alzheimer's mouth. This guy was taught to find this little platform that's sitting right here. And when it was young, but now it can't find it because it's old and has Alzheimer's. What they did is they corrected this IGF-1R pathway, and this is exactly the same genetic background of the same mouse. It actually has phenotype of a neurodegenerative disease, but it moves right to the platform. It's actually pretty normal, and it lives pretty much a normal lifespan. So what we're trying to do is take a protein function. We're trying to protect it with these components that help in synthesis and folding, and there's a series of signaling pathways that drive that, and that, of course, is influenced by your genetic background, what you're born with, by these epigenetic factors, which are really environment, many physiological factors, and of course, what you eat. So this is this system, okay? And the real key here is if we generate the pole, we must maintain it. So I need to now move forward here. What I want to do now is address understanding the balances and imbalances of misfolding disease. I'm going to focus in on one disease, which is cystic fibrosis. This is just a subset of diseases. And this is a loss of function disease, but it's also a gain of toxic function because you lose a lot of interactions. Fact is, the cystic fibrosis protein is a protein that's born in the ER, has to go to the surface, conduct chloride, and if it's doing this, then your lungs and tissues behave normally. We have ways to monitor it the way it shows up on a SDS page, on the ER form and the post-ER form. And the most common mutation in diseases is delta 508, which basically gets born in the ER, but is largely degraded, never makes it out, never goes out to the cell surface. As a consequence, you're in trouble because you're missing this protein. And the fact is, we need to understand these differences. So really, the disease is a sort of a problem of you can't make the protein, you fail to hydrate the surface. As a consequence of that dehydration, this layer becomes compact, it becomes plugged up, and your lungs fail, and so do a number of other organs. And that leads to pathogen invasion and inflammatory pathways, and this is ultimately the endpoint. So turns out this is all regulated by the Proteostasis network. How do we study that? This is our work in collaboration with John Yates and a very talented postdoc, Sandra Panko. And the idea is that we take lung cell models, we immunoprecipitate the protein, and we run it through what's called mass spectroscopy. And then we quantitate the amount of proteins that are interacting with the CFTR protein using what's recovered in the mass spec called spectra. And the fact is, it's a very nice way to get a view of who the protein is talking to, in particular who the mutant is talking to, why it's different from the wild-type protein. And we do that with lung cell lines that are expressing the wild-type protein, the mutant protein, or a drug-treated mutant, to try to understand, does this drug-treated mutant return us to the wild-type state? Okay, the next slide is going to be a little breathtaking, but this is our first view of what this one protein has to see inside a cell. And it looks complicated. So you like to think of your rare disease protein as working by itself. It's not. It's part of a company. And it's an integral part of a company because if you take it out, the company ceases to work. So this is a network, and it's a very special network because we put the CFTR protein at multiple places in here and defined the local interaction partners that make it work. And what we have is already 200 unknown proteins that we don't understand. But what we can do is map out in this pathway. It's born here in the ER. Some of it gets degraded right away, even if it's a wild-type protein, but most of it has to move through the pathway out to the cell surface where it has function, and it actually recirculates at the cell surface to ultimately get degraded by the lysosome. what we've learned with a lot of work that's gone on is that the mutant is stuck right here, and as a consequence, it's completely degraded, so it never sees all these guys. But what we have learned from this sort of network is that perhaps we can push this and get it back to this functional state, and that's largely driven by the biology of proteostasis. And we're going to show, you'll see an example at the end of that. So it's a disconnect between the way the protein wants to fold, and what we have to do is really solve this problem over here. So now what I want to do is just end with restoring the balance in that network. So that one slide was purposely put up there to show you that there are huge differences in the way the wild type and the mutant talks to the rest of the cell. And now the question is, can we manipulate that? In the CF field, we have a drug development pipeline from the Cystic Fibrosis Foundation. Gene therapy was initially a strong focus, which is replacement, and that's still very challenging. It just doesn't seem to be something we can access. Where the success has been is in symptom treatment. All these endpoint aspects of disease, for instance, bacterial infection of the lung, can be treated with a number of drugs, and it's run the half-life or the mean point of failure from 5 years of age to 37 years of age. There's a new class that we're focusing on that are compounds that correct the problem right at the start. So if you put that protein in a corrected form back into the network and it's working, everything else falls into place. You don't have to deal with all these other issues. And we call that a folding for function. And really, it's sort of two approach. One is a chemical stabilizer. You have a mutant protein, you add the chemical, and it gets stabilized. The other approach is what we call a proteostasis regulator. You adjust the proteostasis network. As a consequence of making adjustments in the network, this misfolded step folds. Or we can bring in a new player, put it in place, locks the system down, makes the mutant protein behave. This is the rare disease problem that we've got to really fix. So one thing that we can take advantage of is this is what I showed you for the normal trafficking, but if I drop the temperature on these cells to 30 degrees, this protein goes out just fine. In other words, it's a protein at 37 degrees at physiological temperature doesn't want to fold, but when you drop the temperature, it fits better to the environment because it's not so energetically destabilized. It folds and goes out and looks like wild type. And I just showed that because here's an example of the protein, and we're looking at a lung surface here. At 37 degrees, it's not there, but at 30 degrees, goes right out. And this is what happens when we add one of these proteostasis regulators. Moves it right out to the surface. That's good. That means we can take this rare disease protein, push on it, push on the proteostasis system, and punch it right out to the surface. Where, as this slide shows, we start out in a state where it has this level of channel activity. We add the drug, it has this channel of activity. It responds to the appropriate activators of the channel, and it's inhibited by the appropriate inhibitors of the channel. So we know it did the right thing. Okay, so that's what we're trying to do. And actually, this isn't bad activity considering where we are at the moment. Okay, and we can actually now interrogate that using the work of John Yates and Sandra to ask how did the interactions change. And all this blue is good. That means we restored those interactions, and I'll just show you that very briefly here. And that really means what we're trying to correct are two types of problems. First of all, the wild-type protein doesn't like to see some proteins that the mutant gets hung up on, and if we add the drug, we restore the state. The other type of problem that you have to do in a rare disease is that the wild-type wants to see a protein, the mutant can't see it, but the drug puts it back. And we can do that in parallel using the mass spectrometry. So what we can do is ask the question, we can ask for proteins that bind both the wild type and the mutant. So those aren't interesting because the mutant and the wild type are behaving themselves at that level. There's also proteins that only bind the mutant, and there's proteins that only bind the wild type. When you start out in disease, you look like this. Whole bunch of proteins binding the mutant, a bunch of proteins that only bind the wild type, and very few proteins here in this particular diagram that we selected fall on the line, although there are many proteins that actually fall on the line. Now what happens when we add the drug, and that's shown here. We lose this class jumps up. Look at all the guys that start to fall in the line. So that's just by simply beginning to regulate that proteostasis network. That tells you it's very flexible. We have some room to play here. And we tend to look at this as a partially restored state, which gives a set of interactions that start to look like a wild type. So what we're doing is we're manipulating this system, and now what I want to do in just the last 2 slides is say that because this system is complex, now we have to develop high throughput streams to begin to interrogate it in very precise ways. And one way we do that is develop an assay that we can put in a Multiwell format, and it has a color indicator such that when the drug is added to the plate and the channel moves to the surface, when we assay for the channel activity, normally there would be no channel activity, but when you get channel activity, you see a drop in the curve here. So we call this the extent, and a good rescue goes from here to here. We can also look at the rate, which is a measure of the functionality of that channel. You want that channel to behave itself. It has what we call gating properties, and so we get we enhance the gating properties. This is just one example. This is a compound actually that came from work by a postdoc in my lab, Darren Hutt, and Monica Chaffon, who works in the lab. We've developed this assay in a robust way. And you can see from compounds that came out of this study that was published this year in Nature Chemical Biology, we have the vehicle control here. We have 1x of the compound, 3x of the compound. This is 10x of the compound. And it looks like a 30-degree rescue. Remember that picture of the protein moving out to the surface? That's a really good sign. That looks very promising. Importantly, over here we have this rate. And if you look at the rate, the 30-degree and the 10x value look pretty good. So what I did is, and also I said it correlated with the stabilization of the protein in the cell and its cell surface expression. Okay, so what I did is I focused on this class of molecules. I didn't go into the details, but what about all these other guys? You know, can we manipulate these other pathways? I'm just going to show you one example, and this is pretty cool. So I'm taking now compounds from different parts of the pathway. This is compound A, this is compound B, this is the amount of efficacy you get. Bingo, you put them together and you get synergy. It's a network, folks. We have to correct it multiple places. This protein is still a mutation, but if we teach the pathway at different places to behave itself, it's going to add up. And in some cases, it doesn't. It's just not 2 plus 2, it's the square. It goes, sorry, well, for 2 plus, that's not a good example. Let's say 3 plus 3, okay? It's what we call synergy. If you add these two, you don't get this, and if you look at these rates, you don't get this. And really, we always are very concerned about the difference in the rates here. Okay, just to wrap this up, what I tried to do is emphasize this new system. We call it proteostasis biology. We think it's a new era in beginning to think about how cell biology really works. It's an era we've been missing. We haven't been paying attention to the factors that control the functionality of the fold itself and make the fold. And we think it's a balance. Every day your cells are pushing and pulling on this to be able to handle this balance in a way that keeps you healthy and actually determines your lifespan. And with that, rare disease tends to push you down this path. That's not good. But when you make an adjustment to this, you push it back online again. And really, rare disease is simply evolution out of step. The kids have a mutation that's just a little bit out of reach of the way the system wants to work. So we're hoping with therapeutics that what we can do now is push on the system just a bit, evolve it just a little faster, and keep it at that evolved state. And that way the kids, one, would benefit considerably, but two, perhaps it can learn so that you actually don't have to push all the time. We'll see. That's a real question. It's called epigenetics. Okay, just to end, I need to really acknowledge the people who are my collaborators. I didn't talk about Evan Powers. He's really a great modeler who really impacts the way I think a lot. Jeff Kelly, Andy Dillon, Rick Muramoto are all part of the original sort of thought process that went into being able to consolidate and think about proteostasis. In terms of epigenetics, I didn't get into it too much, but David Herman and Joe Gottesfeld lab at Scripps is a major player. Mass spectrometry was largely the single force by Sandra Pankow in John Yates' lab, and is instrumental for almost all pathway building. We couldn't do anything without this. Scott Becker's in Gerard Manning's lab over at the Salk is our informatics contributor, who's very imaginative and really pushes hard on trying to drive the information level of this. I have many people involved in CF. Darren Hutt and Monica are really the major players, along with Danielle. I didn't show her results, but she's mining that pathway, taking each one of those proteins, knocking it out, asking it how it affects. And then I lead a consortium, are part of, I co-lead a consortium with Eric Sorscher in the CF Foundation. We have a number of collaborators that work together to try to move this forward. And a disclaimer, and also work I didn't show, but we've started a company called Proteostasis Therapeutics, of which I am a co-founder. And a lot of the network work for the proteostasis biology is done by Hui K. Okay, thank you.
Outstanding, Bill. That's very exciting. I have one question before you go. So do you have to, so all the compounds fitting into this general paradigm, do you have to work out a compound for each individual different kind of protein interaction? So for each disease, you would need to know that combination of small molecules or whatever. Can you find groups that ought to act on several different ones?
Right. So that's a really good question, because if you have 6,800 there.
Sorry, back over here.
Good so the question was, do we have to have a compound for each disease? I mean, you got 6,800 rare diseases, but those are rare diseases because of the endpoint phenotype. The folding problem is a basic problem to all of biology. There's only so many ways you can make a fold. So when you learn just by playing the games we're playing, which sort of pathway it falls into, we imagine that one of our therapeutics could correct 40 different diseases, right? But we don't know that yet. That's just speculation. But we're starting at the level of the basic chemistry and biophysics. So we start at the beginning, and we don't try to deal with the endpoint, because if we solve the problem in the beginning, the rest of it's going to fall into place. Okay.
I want to make sure I understand the model. All proteins that work generally are folded to the lowest energy state, or are there examples where it's actually the metastable state, but genetically trapped and actually works that way?
So actually, it's an excellent question. Believe it or not, we're beginning to realize that native state is a picture that X-ray crystallographers give us, is probably the unlikely state in a cell. The protein is actually very dynamic. It mostly exists up above in the metastable state, and actually 30%, some estimate 60% of your proteins are disordered. That's a lot of management. And it's disordered because they have to talk to one another. If you make a little piece of granite, it doesn't merge very well with limestone. But if you grind it up and put it all together, right, you have some flexibility. So we do think there's a lot of disorder going on.
The practical consequence of that is and try to move genes very closely.
That's right. This type of thinking, we're writing reviews on this to try to change the perception by big pharma that little rock that they discovered in an X-ray crystallography experiment is the bottom line. I would predict that probably 90% of the time it's not. They're actually running their screen in the wrong state. So that doesn't negate the utility of that approach. It just says you have to be thoughtful now about what you really have and where you are in the dynamics of the pathway.
One more. Yeah. Oh, you're on.
Oh.
You're on. Yes.
Oh, sorry.
Here you are.
So I read that 20% of mutations is stability.
So, there's, the question is one, only 20% of the proteins are missense, and therefore they would be predicted to be a limited subset of rare diseases. Many are nonsense, i.e. the protein's never made. And then the second question was, if we push on the system, will we cause trouble, global trouble? So the answer to the first question is, you have to back up now. So why are you a missense protein? Well, now you're not recognized either by the transcription or translational system. So it's out of kilter. You need to push on it to maybe make it incorporate amino acid there. We already know you can do that. I talked about protein folding, but I can drive this all the way back to DNA. Your transcriptional and your translational events are the same problem, and they're actually pretty heavily managed by this system. So I would say that nonsense mutations ultimately can be managed, and in the case of CF, we have a corrector, PTC 124, that's in the clinic now, that actually begins to try to address that type of problem. It's a question of what is the system right now ready to handle, what is the capability of the system, and what can we do to push on the capability? And I would argue, the capability of the system is huge. You've just seen it. It just hasn't been exploited. For some reason, biology cuts us off too soon. And maybe that's because there'll be too many of us around. But the fact is, I think we learn to push on those different legs, and we'll be able to actually push most of these forward. But it will be a maintenance problem. Remember, you're going to push a protein out there that still is energetically unstable.